In the next few videos, we're going to go over the metabolic pathways aside from cellular respiration that you need to know for the exam. This video is going to start by going over gluconeogenesis. Gluconeogenesis is the synthesis of glucose from pyruvate and related compounds. In animals, this process occurs primarily in the liver. In many ways, gluconeogenesis is the opposite of glycolysis. Glycolysis takes a molecule of glucose and breaks it down into two pyruvate molecules, whereas gluconeogenesis takes two pyruvate molecules and makes glucose. However, the two processes are not complete opposites of each other, and in this diagram, you can see all the steps in glycolysis and gluconeogenesis. What you'll notice is that 7 of the 10 steps in glycolysis are simply reversed in gluconeogenesis. That means these 7 steps employ the same enzymes in both pathways. However, there are 3 steps in glycolysis that are irreversible because they are exergonic processes and cannot simply be reversed with the same enzymes. And as we're going to see in this video, those 3 steps require a separate set of enzymes to proceed. So here you can see the overall reaction in gluconeogenesis. We take 2 pyruvate molecules, 4 ATP, 2 GTP, 2 NADH, 2 H+, and 2 water to form glucose, 4 ADP, 2 GTP, 6 PI, and 2 NAD+. Overall, gluconeogenesis is an exergonic process. And this might sound interesting because you know that glycolysis is exergonic and releases energy. Gluconeogenesis sounds like the opposite, but is also exergonic. How is that possible? Well, as you notice in this reaction, you require 6 molecules with phosphate bonds that are broken. And you know that when you hydrolyze these phosphate bonds, you're going to release energy. So essentially, gluconeogenesis uses reaction coupling to ATP or GTP hydrolysis to allow gluconeogenesis to be an irreversible exergonic process. All right, so now let's take a look at those 3 irreversible steps. The first step is conversion of pyruvate to phosphoenol pyruvate. As you can see in this diagram, this requires 2 steps. In the first step, pyruvate is converted to oxaloacetate by pyruvate carboxylase, and this requires a molecule of ATP. In the second step, oxaloacetate is reduced to phosphoenol pyruvate by phosphoenol pyruvate carboxykinase. This step requires 1 molecule of GTP. So as you can see, this step is reversed in gluconeogenesis using different enzymes, and it also expends energy in the form of ATP and GTP to occur. Also note that this I'm describing here is only for 1 pyruvate molecule. To make glucose, you need 2 pyruvate molecules converted to phosphoenol pyruvate. So this step really requires 2 ATP and 2 GTP. And then you have several steps that are reversible and don't require special enzymes. The next irreversible step is the conversion of fructose 1,6-bisphosphate to fructose 6-phosphate. In this case, we're simply going to hydrolyze one of the phosphate groups from fructose 1,6-bisphosphate to form fructose 6-phosphate. And this is achieved by the enzyme fructose 1,6-bisphosphatase. Our last irreversible step is the conversion of glucose 6-phosphate to glucose. Here, the phosphate group on glucose 6-phosphate is hydrolyzed, and this produces glucose. And this reaction is catalyzed by glucose 6-phosphatase.